In this video, we are going to study about the difference between spermatogenesis and oogenesis. Spermatogenesis is a formation of sperms and oogenesis is a formation of ovum. We are just studying a difference between both. Spermatogenesis, it takes place in testis. Testis is a male gonad where the sperm's development will take place. So here we can find in the diagram, so this is a testis. It is a testis where a complete process of spermatogenesis will take place. Now oogenesis, it takes place in a female gonad which is called ovary. So this is a ovary where the formation of ova will take place. Next we are going to study about how it differs in the formation. Now when it comes to the process of spermatogenesis and oogenesis, it takes place in 3 steps: multiplication phase, growth phase and maturation phase, which we have studied in a previous video in detail. So now we are going to just see in which phases what all the difference are seen. In the multiplication phase, both spermatogenesis and in oogenesis, the spermatogonium, it undergoes mitosis division. So in the growth phase, we can find a difference where the growth phase of spermatogenesis is short compared to the growth phase of oogenesis. The growth phase of spermatogenesis is short, hence we can say in the amount of cytoplasm present in it and the nucleus remains constant. The size of the primary spermatocyte, we can say it is just 2 times the bigger than the spermatogonia. Whereas when it comes to the oogonia, we can find that the primary spermatocyte, which is in a growth phase, it has more amount of cytoplasm, RNA, ATP, enzymes, and has, due to the accumulation of more amount of nucleoplasm in it, the nucleus also will increase in its size, which is called as germinal vesicle. And even nucleoli also will be bigger in size due to more synthesis of RNA. So in this chart, you can see a schematic representation. This is what is a primary spermatocyte. So here we can find it is a growth phase. This is a growth phase. So here also it is a growth phase where we can find this, the size of this spermatocyte, primary spermatocyte, will be little bit 2 times bigger, whereas when it comes to the primary oocyte, the size will be more larger. We can tell 10 to 15 times larger in its size. Here the deposition will be more. Next we can find that this primary spermatocyte, after the growth phase, the primary spermatocyte will undergo first meiotic division. As soon as the first meiotic division gets over, it produces a secondary spermatocyte. So here we are finding a secondary spermatocytes are seen which are similar in their size. Whereas in case of oogenesis, the primary spermatocyte, it undergoes the first meiotic division. It produces one large secondary oocyte and one small cell which is called as polar body. Next we can find that this secondary spermatocyte, again it undergoes meiosis II division. So this is a secondary spermatocyte. When it undergoes second meiotic division, it is producing 4 spermatids are produced. So each secondary spermatocyte will produce 2 spermatids. So together we can find 4 spermatids are formed. In case of oogenesis, the secondary oocyte, again it is undergoing second meiotic division, but it produces only 1 functional ovum and 1 polar body. Small tiny polar body will be formed. So in case of humans, So after the first polar body is produced, this first polar body will degenerate. So further development does not take place. Further development and division does not take place in case of humans. Whereas in another animals, we can say the first polar body again it divides to form two polar bodies. So next we can find that the spermatogonium. So one spermatogonium, it is going to produce 4 functional spermatozoa. So one spermatogonium will produce 4 sperms, whereas one oogonium will produce only 1 functional ovum will be formed. So the complete process, we can say the spermatogenesis is a continuous process. So after the puberty, the male starts producing the sperms in the testes and it will be continuous throughout his lifetime. Whereas when it comes to the oogenesis, we can say it is a discontinuous process. Why? Because oogenesis will start before birth in the fetal stage. And till the birth stage, we can say development will be taking place. And under oogenesis, we have already studied that it gets arrested in meiosis 1 of prophase 1 and it also gets arrested during meiosis 2 in metaphase stage. So hence we can say oogenesis is a discontinuous process. Next we can say that the spermatogenesis, it is usually completed in the testes. The sperm's development and maturation will take place in the testes. So here these are the seminiferous tubules where the sperm's development will take place. So after the complete development of the sperms, it enters into the epididymis and further transportation will take place. Whereas in case of oogenesis, from the ovary, secondary oocyte is released out. Secondary oocyte will be released out into the fallopian tube. This secondary oocyte will undergo second meiotic division only when there will be a presence of sperm. When the sperm enters into the fallopian tube, once it undergoes fertilization, the further completion of second meiosis will take place. Hence we can say it is a discontinuous process. Next we can say that the sperms are produced continuously from puberty throughout the life. Whereas in case of oogenesis, of course, it starts before birth and ova starts producing after puberty till menopause stage. So we can find a complete list together, whatever I have explained before. All those things you can see in a tabular column. So this is how we can differentiate between spermatogenesis and oogenesis. So at last, after the formation, that is a completion of spermatogenesis, the sperms which are formed, it will be motile, yolkless and minute, microscopic in its nature. Whereas after the complete process of oogenesis, the ovum which is produced will be large in size, rounded structure and it is non-motile. In the next session, we are going to discuss about the difference between sperm and ovum.